My name is Denise Cecil, and I'm the principal scientist at the Cancer Vaccine Institute here at UW-Medicine. At the Cancer Vaccine Institute, we develop cancer vaccines, but we also develop vaccines to treat autoimmunity. The Cancer Vaccine Institute is truly a translational lab, and what that means is that we can take a concept, a vaccine concept, and we can develop that all the way preclinically before we get into people. And then we go to a clinical trial where we're actually testing it in people. And then we had the opportunity to use leftover trial samples to evaluate new hypotheses to make our vaccines even better. So basically we go from bench to bedside, back to bench. To put anything into people, you have to have extensive animal studies to measure safety of anything being put into people. And so the animal model that we use of choice is the mouse. The immune response generated from a mouse is the same mechanism, the immune response generated in a human, so we can translate that from mouse to human. The safety evaluation that we can do in the mouse after the immune response can be translated to humans because the mouse and humans have the same organ systems. And we can also measure efficacy of our vaccine in mouse. There's many mouse models for cancer that have been developed. There's also much known about mouse immunology. And so there's many tools and reagents that we can use for this evaluation. And so in order to cut the timeline down so that we can define a vaccine candidate, we do studies where we can implant under the skin, or if in case it's the breast cancer, into the mammary fat pad of our mice, a tumor cell line that we grew from the spontaneously developing tumor. And so we call that a tumor challenge study. And so we can vaccinate, we can implant tumor, everybody gets the same amount of tumor. Those tumors grow equally well, there's very little variation in those tumor growths, and we can determine if this candidate vaccine is going to work at all. And then we can go into the spontaneous developing tumor to determine efficacy of our vaccinations. In our ovarian cancer model, we use imaging technology. And so basically we have a cell line. I just mentioned those cell lines that from the spontaneously developing tumor. That cell line expresses a construct that bioluminescence when it sees its right enzyme. And this is the same type of bioluminescence that you see in fireflies. And so this is called a firefly luciferase construct. And the cell lines express that. We inject the cell line into the peritoneum of the mice to mimic where ovarian cancer would spread. And then every 2 weeks, we're able to inject the enzyme luciferase, and that the tumor cells will bioluminesce for about 10 to 20 minutes. And we're able to, under isoflurane, monitor those mice every 2 weeks, and we can monitor that tumor growth as they develop. All of our technical staff are validated and cross-trained. And we believe that this rigor allows us to use fewer mice per group because we're not relying on the variation in technical differences. We're just looking at a variation in the animal's responses.